Olá, buenos dias a todas e
Hello everyone,
todos.
good morning everyone,
Bem-vindos a mais
and
esta
welcome
edição
to one more
do
edition of
programa de
this
webinários
program of
de RedETSA.
webinars of
Saludo a
RedETSA.
todos os membros da
Greetings to all the
RedETSA
members of
e
this
nossos
network
invitados
and
que
several of the
permanecem lá dentro,
guests
que
that are not part of this network
participam
but
desse
participating
programa de
in
webinário.
this
Muitíssimas graças por sua
series of
participação.
webinars. Thank you very much for your participation.
Já desde já agradeço sempre a minha equipe
From now, I'm already
de
thanking the
pesquisa,
team that is
de
organizing
voluntários,
the
especialmente a Marlene
webinars,
Holman,
especially Marlene Holman,
que todos conhecem,
that I believe that you all
Vale
know them,
Salomão
Vale
e a
Caruto, Vale Salomon,
Lisete
and
Nunes também,
Seti
com
Nunez
todo
as
o
well,
equipe que trabalha muito
with
para
the team
manter
that is
esse
working
programa
hard to
muito,
keep alive
muito
this
exitoso,
program, a very
esse
active,
programa
very
de
successful,
webinários.
this
E hoje
webinar
teremos
program.
uma edição especial,
Today we had a special
porque
edition
estamos organizando em
because we
conjunto
are
com a Health
organizing
Technology
together
Assessment
with
International, HTAI.
Health Technology Assessment
Esse treinamento
International,
conjunto não é o primeiro que estamos
HTAI. It's not the
Esse
first
é
one that we
até
do together. I
terceiro
believe that this is about the
desse
third one
programa,
that we do together about
uma
this
atividade
program
conjunta.
with
Penúltimo
HTAI.
foi recém,
The last one was very
iniciação de
recent
processos
about the
eletrônicos,
involvement of
muito exitoso
patient
também.
with the use of new technologies, very successful as well.
Teremos um tema
And
também
now we had something very
sobre
important
evaluação
as
de
well
tecnologias sanitárias
about
aplicadas
evaluation
nos
of
IVDs,
applied health
laboratórios de
technologies in
diagnósticos
IVDs,
in vitro, não?
diagnostics,
E
IV tests.
queria mencionar
And I wanted
que
to
muitos
mention,
aqui sabem que o secretariado de la red hoje
so I believe that you know
foi
that the
reconhecido por novo
Secretariat of
departamento.
the network
Historicamente, o
has a new
secretariado sempre esteve
department.
na unidade de
Historically, this
tecnologias
secretariat
radiológicas.
is always the
Simultaneamente,
health
esse departamento
technologies
tem
of PAHO,
departamento
and this has
que é
become
o
a
departamento de
department
inovação e
now that is
acesso às
the
tecnologias sanitárias.
Department of Innovation
E
and Access to
nessa
Health
unidade,
Technologies. And our unit,
eu estou
in
responsável,
this unit, I
a
am
unidade
responsible right
de
now,
acesso a
the
tecnologias
unit of
sanitárias.
access to health
Então, se tem um
technologies, well,
secretariado
has now
desta
a
área.
secretariat
E
of this network.
estamos trabalhamos vários
And we are
temas.
working in
Evaluação de
several
tecnologias é um dos
topics.
principais
Evaluation
eixos
of
de
technologies,
operação
one of the
desse
main
departamento de nossa
axes of
unidade.
cooperations
Trabalhamos
of
com
this
vários temas de
department of our unit.
acesso,
We work with different things.
estado de
Policies
tecnologias
about access,
essenciais
the
e outras
status of
tecnologias.
technologies, essential
Trabalhamos
medicines, and other
operacional
technologies.
também de tecnologias. Trabalhamos com
We
vários
also
temas.
work with national technologies
Nessa
and several
unidade,
other
nosso
topics. And in
secretariado está
this unit, we are
avançando
each time
no
more
tema de
moving
diagnóstico. Esse é um
forward with
desafio,
everything that has
porque
to do with
por
diagnosis.
muito
It
tempo
is
temos
quite a
priorizado,
challenge because for
principalmente medicamentos,
quite some time we have
essa atividade
prioritized
central,
medications
os medicamentos
in a
estão
more
priorizados
general
nas
way. The
pandemias, né?
medications have
E
been
pouco a pouco,
prioritized in different ways, and little
devido à
by little,
pandemia,
I believe that
sido wake-up call
pandemics has been a
para muitos
wake-up
temas
call for many of these topics
dizer também que outras tecnologias também são
about
prioritárias,
having
como
other
dispositivos médicos,
technologies
incluindo
as medical devices,
Então,
including
cada vez mais estamos
diagnosis.
crescendo
And each time we are moving
também
forward,
os temas
each time
relacionados ao
more
acesso a
about these
diagnósticos
traditional
e hoje
topics of
especificamente
diagnosis and access to diagnosis. And
trabalhamos
now
com o tema de
specifically,
evaluação de
we
tecnologias
have the
aplicadas no
evaluation
diagnóstico.
of
Houve
applied
bastante
technologies to
progressão
diagnosis. We had some recent
tecnológicas
progress
nos últimos anos em termos
in the
diagnósticos.
last
Um foi
years
desenvolver
about
alguns
diagnosis. One
status
was the development
diagnósticos
of the
essenciais pela
essential
OMS
diagnosis
e alguns
status
países.
established by WHO and some other countries.
Nosso equipo está apoiando,
Our
trabalha muito em
team
conjunto
is
com a OMS nesse
supporting
sentido, já nos
a
traduzido
lot of this work
publicações,
with the
listado,
WHO.
próprios
We have
estados
translated several of
diagnósticos
these
essenciais,
lists
também
themselves of
estamos
this
trabalhando também com
essential diagnosis and another
esse listado de referência
another article about updating this list of reference around the globe for the different countries. And especially, I don't know if we have here with us a colleague from Honduras, but especially with Honduras, we are supporting this first official list, first the national list of essential diagnoses adapted from the global state, what we called Essential Diagnostics List, EDL, of the WHO, World Health Organization. So I wanted to mention this context. I believe that it's pretty important. Also insist and highlight this collaboration that we have between RedETSA, PAHO, and HTAI. As I mentioned, some of these webinars, we have RedETSA and HTAI. We have an agreement of understanding uh, signed very recently on with a series of activities that are prepared. And as you know, we are organizing this in collaboration, this encounter of Redets this year. As you will know, it's going to be the same week. We are organizing joint activities between HTAI and Redets for the next meeting that we're going to have in a couple of weeks in Buenos Aires, Argentina, and where we are going to have in there back to back, almost one after the other, this encounter of RedETSA and this encounter of HTAI. It's important to mention that on top of it's not per chance, of course, that we have been working together with HTAI to generate this encounter in the context, of course, of HTAI, creating joint workshops. We have always been very close working with HTAI. So I wanted to thank also about what we're going to use there, the different spaces, workshops that were agreed on for the Congress, a series of joint activities in the context of this encounter of HTAI. So we are extremely happy to be able to have, for the second time in history, an HTAI encounter in Latin America after having the great honor of having in Brazil the first one in 2011. Now we are going to have the second one, a second Latin American meeting of HTAI, and having the REDETS meeting in the same context in the beautiful city of Buenos Aires, Argentina. So I wanted to record this and thank the collaboration with HTAI. I think that time's up. Going to close here very quickly this opening. I'd like to thank once again our different speakers and panelists that we have today, starting with Stephanie, that is our colleague here from the team, and our consultant, the specialists in diagnosis. Especially, that's why we invited here to moderate this session. And also Anastasia from the United Kingdom, and the co-chair of this group of interest of medical devices of HTAI. Thank you very much, Anastasia, for this. And we are going to respond to our panelists, our speakers for today. I'd like to thank Federico from Argentina, Rosa Maria from Spain, and Christopher from the UK, that later, obviously, are going to provide their introductions. They are going to be introduced actually by the moderators that are going to be Anastasia and Stephanie. So once again, thank you very much for this participation in this session. We have uh, several people, about 150 people connected, and I wanted to thank once again for this participation and to say that we have interpretation available for English and Spanish. We have the recording as well. It's important for you all to know that we're recording this session and that then this is going to be available on our website of RedETSA, and it's something that we're going to send through email, this link with the recording. So thank you once again, and I'm going to give the floor right away to our moderator, Anastasia Tokida. Thank you very much.
Thank you. Thank you everyone for registering today for the webinar and choosing to spend your Friday afternoon or morning learning more about HTA of diagnostics. I also want to thank the organizers for asking me to co-moderate this along with Stephanie. I'm program director for guidance delivery for health tech at NICE. So my day-to-day job involves overseeing our portfolio of guidance, specifically on medical devices, diagnostics, AI, and digital. Diagnostics is a very interesting and ever-growing part of our portfolio at NICE. Obviously, it started early on with in vitro diagnostics, and now with more of the digital and AI stuff, we see an increase in imaging-based diagnostics, AI-based diagnostics, and we know that in medtech and especially for diagnostics, there are sometimes specific considerations and requirements that we need to keep in mind when we evaluate this type of technologies. And obviously, our panelists will explain what that looks like and give us a flavor of the methods and the state of the art. Today, I want to remind everyone that after the presentations, we will have a Q&A session. So please do write your questions in the Q&A function by clicking at the bottom of the screen. And I'll pass on to Stephanie to introduce herself.
Thank you. Thank you very much, Anastasia. I'm going to switch now to English. Muchas gracias a todos los participantes, los panelistas.
Thank you very much to all the participants, the panelists, and HDAI for your collaboration in this beautiful event, and all the organization team for organizing this team. My name is Stephanie Wimet, international consultant, the IMT Department of Tahoe in Washington, and I work in everything that has to do with access to health technologies, more specifically the diagnosis tests, the in vitro diagnosis tests. And it's a great pleasure to be with you today, moderating this event together with Anastasia about a topic that is very relevant and without a doubt. We are going to have very interesting and pertinent conversations, and this is going to carry on forward after this webinar, of course. And as Anastasia reminded you, please write your questions in Q&A. You can write them in English or in Spanish, whichever you prefer. And we are going to take some time after the session so we can have a group discussion about it. I give the floor back to Anastasia so she can introduce the first speaker. Thank you.
Thank you, Stephanie. So our first presenter for the day is Professor Chris Hyde from the University of Exeter here in the UK, with a long history of looking at methods for STA, for diagnostics specifically. Chris will present an overview of STA methodologies for diagnostics test. I believe that work represents the MRC test project group findings. Over to you, Chris.
Hello, everybody. Let me share my slides. Right. Hopefully you can all see that.
Yeah, we can. Thank you, Chris.
So I'm going to talk to you about a project which we are still doing at the moment, but this represents one of the early outputs of what we call the MRC test project. And it represents a publication called Health Technology Assessment of Diagnostic Tests, a state-of-the-art review of methods guidance from international organizations. So there is a full paper on this already. So I'm not going to spend too much time going through the detail of the paper, because otherwise that would be a very long presentation. So just to acknowledge the other centers who are involved in this, particularly Lavinia, who was the first author of the paper. She's now working at York, but used to work in Birmingham. And there are groups of people who work in Birmingham, and Sue, who's a medical imaging specialist who works in London, and myself and a group of people who work in Exeter. We have lots of acknowledgements of the HTA organizations who we liaised with and who provided us with lots of feedback on whether we'd understood their guidance correctly, and also to our steering group who commented on the drafts of the paper. And just to acknowledge that the work was funded by the Medical Research Council and that none of the authors on the paper have any conflicts of interest to declare. As I said, I'm not going to go through the paper in detail. There may be perhaps in the questions, if there are things which I've skipped over, which you would like to talk to me about, those can come up in the questions, but I just want to say a little bit about the methods because some of them are quite detailed. I want to say a little bit more about some of the headline conclusions, to expand those a little bit, and obviously to give an opportunity for questions and discussion. So I think we've covered this ground a little bit already, but just to repeat, new medical tests are rapidly expanding and increasingly expensive. The sorts of things which cause everybody problems, not just NICE, but I think NICE has been at the forefront of, been on the receiving end of all of these things: companion diagnostics, genetic tests, artificial intelligence, and applications are some of the things which are causing HTA organizations particular challenges. The fact that we haven't really got to the bottom of this is illustrated by the fact that there continues to be controversy about tests for COVID. So if ever there was an example where we needed rapid and authoritative advice on which tests and which what the effectiveness of those tests were, it was COVID, and I'm not sure we really achieved that. And I think the particular challenges for the evaluation of tests, particularly IVDs, is that they don't exert their influence directly on patient outcomes. They do that indirectly, so that represents a challenge for evaluation. There's a widely held assumption that tests mainly bring about benefit through improved accuracy of diagnosis, and that's almost certainly incorrect. It may be true in some diagnostic tests, but for the vast majority, the object of new tests is not necessarily to bring about improved accuracy. It's to maintain accuracy, perhaps by reducing the time to get a test result to a patient. And that current methods for tests are based on the extension of HTA methods for therapeutics, which means that they may not be specifically adapted to the challenges of diagnostics. And there has been virtually no investment in methods research for HTAs of tests, because I've been trying to get it for the last 20 years, and it's extremely difficult to come by. So that definitely represents a challenge in terms of defining what are the best methods. So I'm now talking about the paper that we did. So how would I describe the piece of research that we did? I would call it a document review, and the method of analysis was qualitative. We were exhaustive, and I would say also exhausted by the time that we got to the end of the this particular piece of work. We made considerable efforts to identify all potential HTA organizations who might be offering guidance on how to do diagnostic HTAs. And we also made considerable efforts to abstract and agree what each of the main organizations said about predefined aspects of the HTA process. We did duplicate assessment, so they were certainly all of the abstracted information was done at least in duplicate and sometimes in triplicate and sometimes in consensus. And I would say that in terms of reflection, that synthesis was probably the most difficult component of what we did. Here's just one of the diagrams from the paper, which gives you some idea about the exhaustiveness of the identification of the process. You can see that there are, if you didn't already realize, there are quite a large number of HTA organizations across the world. And of those, only a minority either have a responsibility to look at tests and then to say something about it in their guidance. So we actually boiled down, there were, of the 351 potential organizations, we identified 41 which were actually providing some guidance or said something about diagnostic test evaluation. And of those, we used 7 main ones to identify our themes. And we checked whether those themes were consistent with the stage 2 organizations, which is the 34. So what were those main themes? So these are the themes in the analysis. They were, well, first of all, the elucidation of claims of test benefits, the process by which that was done. Secondly, the attitude to direct and indirect evidence of clinical effectiveness, including evidence linkage. Thirdly, searching. Fourthly, quality assessment. And fifthly, health economic evaluation. So these are the things which we comment about in the paper. And I'm not really going to touch on a number of those, but I will just drill down into the first 2, because I think those are things which you might find of particular interest when you're trying to do your own health technology assessment of IVDs. So here's quite a complicated diagram which appears in the paper, and I just thought I'd take a little bit of time to explain it. And there's a further diagram dealing with the use of direct and indirect evidence. And you might say, oh, Chris, what do you mean by there's variation in how we deal with claims of tests? So the green box here represents what the majority of HTA organizations, they all said they explicitly identify test claims during the scoping phase. But after that, we get some divergence in what people do. And in some times, people then follow that identification through into categorizing those claims. And sometimes they then use that in turn to prioritize the outcomes, which are felt to be most important. And then they also then take it further to feed that into the health economic methods. Whereas other organizations perhaps don't go quite as far along that line, perhaps stopping at the point at which not incorporating it into health economic models. That's what that grey box suggests. And in the scoping phase, they may only identify outcomes without prioritizing them. And indeed, in one or two cases, we found that test claims were not explicitly considered at all. And there was just a general scoping exercise, which was then used to identify outcomes. So there is quite a lot of variation, even with this very simple and basic step in the HTA process of thinking about, well, what are the important things which might happen as a result of introducing a new diagnostic test, and how those are incorporated into the HTA process. And I would say the ideal would be, if you can achieve it, although it's sometimes easier said than done, is to prioritize health outcomes and to make sure that that prioritization is incorporated into health economic models, health economic methods, health economic evaluation. But that's a personal preference, and people might say, well, there are practical constraints to whether you can actually do that in practice. So here is the second diagram from the results, and this concerns the theme about preference for direct evidence. And you can see here, again, the green represents what the majority of HTA organizations in our stage 1 identified. The majority preferred direct evidence. The majority felt that direct evidence was unlikely to be available and said that they would consider indirect evidence. But sometimes we were unable to identify specifically what people were going to do in the absence of indirect or of direct evidence. And the thing which I think is particularly worrying, given that there are increasingly like to be the existence of both direct and indirect evidence. So direct evidence, I mean end-to-end studies or randomized controlled trials where you introduce the technology and then look at the difference in outcomes in a piece of primary research, as opposed to indirect evidence, which is a sort of thing where we build an argument containing both information about the accuracy of the test and then information about whether what the effectiveness of treatment is. So that's what I categorize as indirect evidence. And we were quite interested, or personally, I was quite interested in the fact that none of the HTA organizations in stage 1 said that they didn't really have a preference for whether they preferred direct evidence. And I think there is a number of bits of methodological research which suggests that RCTs in particular may not necessarily be the least biased way of identifying whether there's improvement in patient outcomes if you introduce a new test. Personally, I would say that we shouldn't have a preference for direct and evidence, and we should, in our HTAs, consider both direct and indirect evidence, and we should make sure that we have methods to combine those two types of evidence. So here are the conclusions that we had. So this is of the paper itself. There were areas where there was consensus, and interesting, many of the HTA methods guidance documents said quite a lot about dealing with test accuracy, provided some examples of good practice in terms of systematically reviewing tests of test accuracy, although there are many areas where we struggle, particularly in the area of meta-analysis and dealing with heterogeneity, but at least there was consistency across the HTA organization. But the thing which we thought was really interesting, and hopefully something which you will be able to reflect on as a group, is although we spend a lot of time talking about how to deal with test accuracy evidence, we spend very little time, we acknowledge widely accuracy is not really a sufficient evidence base for test evaluation. So if we only had evidence on test accuracy, we would probably be comfortable recommending that we would want to have some information about what the effect of the test was and the misclassification of those tests were. And the two areas which we thought were important from a methodological point of view were this integration of direct and indirect evidence and standardizing approaches to evidence linkage. And that, by that I mean the combination of evidence on accuracy with the combination of evidence on how effective treatment is. So my advice to you, particularly as if you're beginning to develop, is to keep it simple. I would say it's incredibly important to concentrate on understanding the claims of the new test. You'll probably find that defining current practice will be more challenging than defining the new clinical pathway with the new test in. That's certainly been my experience. When you're looking specifically for once you've identified your claims, then you can look specifically for evidence that confirms or refutes your claims. This helps you focus in on perhaps the key aspects of benefit and helps you possibly simplify the HTA process. But once you've done that, you must consider whether there are any aspects of safety which have not been recognized in the claims for the new test. So perhaps there might be a trade-off, let's see, between time to achieve a quicker diagnosis with the accuracy of the test. The final thing I would emphasize is that understanding the balance, or balancing harms and benefits, the pros and cons, remains a highly subjective process. So we need to recognize that when we're making our decisions on the introduction of new tests or the reimbursement of tests. But I think in the current state of knowledge, that our ability to quantify the harms and benefits may not help us very much with that subjectivity at the moment. Possibly if I were a health economist, I would point to you that the cost effectiveness modeling is adequate for that, but I think our work suggests that that is probably not the case, that the cost effectiveness modeling that we're used to doing when we're looking at therapeutics may not work quite as effectively in the diagnostic space. I think that's it. I hope I haven't run over too much.
That was great. Thank you. Thank you very much, Chris, for your presentation. We will now move on to our next panelist, who is Federico Augustowski, the Director of the Department of Health Technology Assessment and Health Economics at the Institute of Clinical and Health Effectiveness in Argentina, and who will present to us their work on a value framework for diagnostic technologies.
Excellent. Thank you, Anastasia. I will switch to Spanish. Many of my slides are in English, but I will switch to Spanish, and I understand that there is translation in case you need it. Bueno, un gusto estar aquí. Gracias.
All right, a great pleasure to be here. Thank you for this invite of RedETSA and HTAI. And we are going to start with telling you a little bit more about our experience in trying to move forward with a value framework of diagnostic tests. in a couple of projects that we were involved in a couple of years. The idea is to tell you, with the time I have available, I have 15 minutes. So the idea is to tell you especially about a project that we did in Latin America that Vania was involved in and was part of this strategic committee that was trying to propose a value framework for diagnostic tests in Latin America focused on the decision-making of coverage, reimbursement in the world of ETS in Latin America with the participation of several of the RedETSA organizations and the people from there. We're going to focus a little bit on talking about this project, and if time allows it, then I can tell you also that thanks to this project, they called us later to adapt this value framework of Latin America in a context of diagnosis tests that are more specific, generic, that are next generation sequencing, and comprehensive generic profiling within the European context. In regards to oncology, for instance, is a project that is dealt with the International School of Economics, and the Q&A sessions at the end of this session with all the panelists. So just as Christoph just focused in a couple of strokes of this huge project that he mentioned about the MRC diagnostics, I am going to put in here the two papers that we published about this initiative of Latin American value frameworks. So you're going to get more details, and any one of you that is interested on getting more details about these projects. I'd also like to thank all the people involved from Mike Raymond from the York University, the EX team, and the steering committee. This project had two stages. One stage more about development of the value framework that we are going to tell you, and another stage more of how easy it is to implement this, how feasible, how ready for prime time it is, how ready this would be for the different agencies in the region. So in each one of the stages, we had this group of people that was with us in this steering committee. So the first part of this project, what we did was, I'm going to tell you a little bit more of detail, but it was a general literature check of the current status of literature to find value frameworks, be it generics or specific for diagnostic tests, synthesizing the different domains, the dimensions that we were proposing or evaluating within these frameworks, and then after that provide or contribute this for a group of stakeholders or in around 20 people of Latin America, between 20 or 30 in each one of these rounds on the world of the diagnostic tests or this ETS and the world of the regulatory part to try to separate apples from oranges and see which are the most important criteria that have to be in an evaluation and which shouldn't be in these kinds of assessments, trying to categorize these according to importance. And in the second round, what we did is to generate, so this value framework would have operational tools for the agencies that would like to implement it and use it. So as we were saying, a manual for researchers as well as, I don't know, decision makers. We generated some documents about this, applying this new value framework. And we have a couple of educational activities, these courses within the EX campus, free of charge and free of access, so whomever wants can internalize these topics. I didn't mention that this project, round 1 and round 2, was financed by a great independent part of the Roche firm This about the members that we had in both rounds. Around 9 members of 10 countries of Latin America participated. The first round was pre-pandemic, was face-to-face in Buenos Aires. We discussed the framework, and the second one was post-pandemics. So this was through the great window of Zoom that we're using right now today. And in the first paper, where we described a little bit about the first stage, what I was saying, well, first we did literature studies to identify value frameworks, something similar to what Christopher mentioned, and they did a more thematic qualitative analysis. We wanted to try to define the domains and ponder the importance that people would give to the different domains. So we had a couple of quantitative components that were additional to the qualitative. So we proposed a list of initial criteria. Then in the second stage, we categorized the criteria about which are the high-level domains, and a couple of them have sub-criteria, which criteria belong to each one of the mother criteria there. That this was done together with the participation of the stakeholders, and this, after we had the list of big criteria and small criteria or sub-criteria, we had a couple of rounds of consensus to evaluate the importance of these criteria, evaluating diagnostic tests for an agency of ITS of HTA, and then we had a qualitative. We are not going to extend too much due to time frame to identify barriers and stakeholders in the region to move on with the more formal evaluation that, as Chris said, they are quite behind in a way compared to the therapeutical interventions. And finally, generate a proposal for a framework and dissemination as well, so the information can be spread. And when we checked this literature, this was 20 value framework in the agencies. It was not only the frameworks of the HTAs, but it was also about scientific associations, not only the world of HTAs, but the synthesis and curating the criteria and sub-criteria of this value framework. We had 28 criteria with 21 sub-criteria. This is one of the tables of the paper. That's where we ask the stakeholders in Latin America to say which criteria are substantial, high importance, moderate importance, or low importance. For instance, the clinical benefit connected to the use of a test and the performance, the operational characteristics, accuracy. Most of them, about 9 of 10, consider this essential, same as the quality of scientific evidence, security aspects. In this first round, we categorized the dimensions of the potential criteria of our value framework that was the oven, baking, with 31 total responses between all the different stakeholders of the team of work.
Then we conducted a second round to be able to show the results of the first round to see if people would agree with what was on the first round, or if there was a difference of opinion, or anything like that, etc. So to try and like rank the importance of each of these criterion. Down here, you can see it was a face-to-face encounter back those days. And very close to where the HTA conference is going to be held in Puerto Madero, Buenos Aires. That's where we met. So then finally, also in our website, in EX, in the value framework, both in paper 1 and paper 2, and academic materials and conveying materials, in case you're interested. Also, the 5 essential criteria, which will be the clinical benefit, the performance, the certainty, scientific evidence, and also some other importance, either high or middle importance criteria, and low importance as well, and the plus of innovation, or the component of innovation. Which oftentimes the producing industry considers it of high importance. And we also have the definition for each of these criteria. It's in English, Spanish, and Portuguese, as with any material else, so that you can take a look if you're interested. Then on the second part of this project, this second round had to do a little bit more of how to make this usable in a real-world setting. Is it a usable value framework, either potentially or not? Can it be implemented? So what we did was to follow a methodological guide. So if we were a researcher and wanted to bring an HTS document incorporating the various dimensions of the value framework, and also some other recommendations we give is that we don't only have this list of criteria. Some of them are high or low or moderate importance, but also how to decide on which one to use, if it's dependent of a country, if it's related to an agency, etc. So it's not a canned-in criteria. You have to check and see what kind of criteria does this work together with, and create your own pathway, your own journey. So we created a methodological manual for researchers in case they wanted to use the value framework and each of those sets of criteria to assess them with the new diagnostic technologies. And so we checked on what kind of tools are being used today to assess this criteria. How could it be evaluated, the sustainability, the organizational impact, etc. And so we tried to provide recommendations, the simplest and minimum recommendations of what would be the minimum way to apply this criteria, or oftentimes using more thorough, more complete technologies. Then we started with the piloting and chose 2 technologies to be able to draft an HTS document, and we created this rapid HTA document to create a value document, and included a little bit more of the feedback and the methodological guide into it. And then after this engagement with stakeholders, what we decided to do was tell them, okay, if you receive a document such as this in your agency, then it would be useful. It would help you in decision making to check what are the the boundaries, limitations, or constraints that you see that are there for the implementation of these value framework. What more qualitative sort of boundaries are there? What is it missing? What is it good in this framework currently? So this is a little bit of the image in the paper of all these second round phases of the project. Here are the links for the technical manuals, both for researchers as for decision makers. An example of the document we came out with that at that time was for HPV testing, the genetic HPV testing, which sometimes are self-managed by women. And here are the 15 dimensions, and a little bit of the categories of how good or bad the evidence was in favor or against these 15 dimensions. I'd also told you about these courses that had more than 300 students in the region and that are still open in case you want to come closer to the diagnostic testing and the HTAs. And this is a little bit of a summary slide and how this panel of 20 people that study HTAs in the region reacted to it, whether it is useful, whether it improves decision making according to the current way to do it, that is not customizing diagnostic testing, how much training effort will it require in the region. It's going to demand some effort, the usefulness of it, the relevance and implementability. There was good motivation from these participants. They found it very easy to use and to understand. Okay, and so this project of European oncology is one that we're going to look at later on, but you'll have also papers in the material and the recording. And I go straight to conclusions, as reaffirming a bit of what was exposed by Michael during the first session. Diagnostic tests are a little bit more complex bugs that have their own differential challenges, different therapeutic interventions. We show what can be done to be able to make them easier to address, and then the tools for the various dimensions that are relevant to them, and also focusing on the higher value dimensions as perceived by healthcare systems. But well, this is the end of my presentation, and then we'll see you later during the Q&A.
Thank you very much, Federico, for your presentation. I will now pass on to Stephanie to introduce our next two panelists.
Thank you very much, Christopher. Muchas gracias, Federico, por su presentación.
Thank you so much, Christopher, for your presentation. You have brought to us very complimentary presentations about the breakthroughs and the progress pertaining diagnostic technologies assessments, the HDAs. Now we're going to continue with our next panel. We'll have the participation of two experts who will be sharing with us about case studies. First, we have Rosa Maria Huidaco. She's the Director of Assessment in the Agency of Quality and Assessment from Catalonia. So, Rosa Maria, without further ado, go ahead.
Thank you very much. I'm going to present now this case study. You see the full screen?
Sí, perfecto.
Vale, pues yo voy a hacer como ha hecho Federico y la presentación está en
So as
inglés.
Federico did it, the presentation is in English, but I am going to do it in Spanish. So I'm going to introduce this case study, which is real. This is a report we conducted last year about the diagnostic usefulness in a liquid biopsy for the analysis, more specifically in this case, and the circulating tumor DNA using this type of technology of next generation sequencing, and more specifically for the initial diagnosis and screening of breast, lung, and colorectal cancer. This request came from the Catalonian Service of Healthcare, most particularly from the new management of criticism healthcare, and they wanted to look at critical care, and they wanted to see if they could incorporate these tests in cancer diagnosis, and it was very much focusing on trying to see initially what was the diagnostic usefulness in the context of this situation. And so what we had seen, what they had seen, is that there had been sufficient progress in terms of technology of NGS to see if it could analyze the DNS and ctDNAs from liquid biopsies, and for what some experts have said that it could have potential applications in the various phases of cancer, both in the initial or diagnostic screening, and also in trying to profile the different genetic mutations in these tumors and to monitor the response to treatment or to spot the minimum response or to select what were the most appropriate treatments based precisely in the detection of this genetic disturbances. And different to what is found in tissue biopsies, what we could find and what we see still is that liquid biopsies are minimally invasive procedures and that they enable to have a good collection of relevant cellular components to be able to assess the genomic profile in real time. And on the other hand, some other of the advantages is that they didn't need any more invasive or costly procedures. They could save money that way. And what was being used usually to be able to assess with liquid biopsy, the samples being used most frequently were plasma that were obtained from blood. So then having all of that context in mind, and most specifically, they asked from the management. Oh, and before I mention that, I've said that what it was going to be evaluated would be the ctDNA, but this is one of the complexities of liquid biopsies, and it is that you are able to detect several components, but diagnostic tests specialize in those components. That's why when they were telling us to analyze the liquid biopsy, okay, but the liquid biopsy needs to analyze what specifically? And it was very important to determine that because you're able to understand how to detect tumors in blood, free DNA, which includes in our interest the circulating tumor DNA, and also the free RNA, proteins, metabolites, or extracellular vesicles. So when you were saying analyze the liquid biopsy, you'd have to specify what is it that you needed to analyze. And if you could come up with an answer to what is it that you would find. So you got to that specific question for us to be able to assess the diagnostic usefulness and utility of liquid biopsy. And so in that context, what we did is we broke down in several objectives. So the first thing is they asked for us to provide available evidence and diagnostic utility of ctDNA using this technique of NGS in breast, lung, and colorectal cancer, and also to be able to specifically determine in a kind of stage of cancer patients we're at. At the same time, they also wanted specific information of panels that were already being marketed or being used already in some centers. Some other objectives were also to identify what were the recommendations from these scientific societies in regards to the use of liquid biopsy as first option in breast, lung, and colorectal cancer. And on the other hand, they also asked for us to create a protocol of what would be this budget impact by introducing this type of technology. And for this presentation, I'm not going to address this sub-objective for too long. And since we have a very limited time to respond, what we did is we brought a revision with the Cochrane method. And what we did is with the information of these tests, where they wanted information from, to have a narrative review, and also with a recommendation of the scientific associations. So what happened is we didn't find systematic reviews that could talk about the diagnostic, the screening of liquid biopsy, specifically using ctDNA with the generation sequences. So what we did is, as Chris had said earlier, we had to go to the indirect evidence. So we brought up to them, we could find systematic reviews And if we could do it to check the molecular profile of these tumors, then that would be useful. So we focused this report as a diagnostic utility in this kind of indications. And then we found a systematic review for advanced breast cancer that would specifically analyze this gene, and that there was a certain sensitivity that could be considered high, but this analysis of this gene would only apply to a subpopulation, a specific subpopulation of breast cancer patients, where you could find one of the limitations here. For big cell lung cancer, you would also find the sensitivity that was very specific. And in this case, you would also find some other parameters, such as the positive percent agreement, the PPA. So here we find they give you sensitivity, but they also give you some other kind of metrics for diagnostic utility. And so we found a report talking specifically about this mutation analysis with a sensitivity of 0.78%, 78%, a specificity of 85%. And this is a mutation that in that report was only addressing this particular mutation. In the case of colorectal cancer, we found a systematic review that would address the RAS genes in terms of diagnostic utility, but we didn't find that it would be applied to different stages of cancer, because they were talking about advanced cancer, but they wouldn't specify where. And in the case of panel results, we contacted the companies also via web search, but what we found in this report, or we're able to provide in this report, was a chart where we describe the various features, talking about the use intention, the identification, what is the regulatory status, the genes being covered, the types of the syndromes that were being detected, the kind of sample being analyzed, and what were the results provided. So you're going to be able to see that from the 12 panels that in the end we used top 9 because there were 2 companies where we find that these 2 panels had differentiated only one single panel. So we didn't find any one panel that would look into a useful intention for screening or the initial diagnostic of cancer. And so what we were finding was a lot of variability, and I'm not going to take too long on that because I'm over my time. But I'm going to focus on this, on the great heterogeneity, and that there were 7 panels providing info about sensitivity, but only 4 were giving us info about specificity. So there was also about predictable values, negative or positive. There were very few panels that would report that. Only 2 of them would provide some other kinds of metrics, as I said earlier, and the PPAs. And at the same time, the results that we were finding, some of them were based in selections, in variants, so there was a big heterogeneity. And in terms of the results from scientific societies, we found that none, neither the Spanish, European, or North American, would accept the use of liquid biopsy for an early diagnosis or cancer screening right now, but they do for other types of circumstances to guide treatment or stuff like that, but not to bring an early diagnosis. So you can find here that there are big restrictions and limitations for this report. We find that we haven't specifically found systematic reviews addressing this, but it doesn't mean that there are none. But since we focused on this kind of reports, we weren't able to find systematic reviews on the matter. And so what we did through indirect evidence. And one of the limitations of these systematic reviews was that they did not make a difference in the technologies used to analyze the ctDNA, nor differentiating the cancer stage where this technology was being applied. In terms of limitations in the comparing elements, some of them were about tissue, but they were not specifying the techniques for this kind of biopsies, nor the liquid biopsy allows to identify the molecular heterogeneity that it provides. These are the conclusions. So the systematic review evidence for diagnostic utility took us to indirect ctDNA. We found that the methodological quality was low. And I'm not going to take much longer on this. And what I do want to get to was this part. What I wanted to share, which Federico and Christopher had referred to as well, is not a great heterogeneity of results and the samples and the metrics used and the characteristics of patients. What this does is it makes it hard to find a good interpretation of results, and at the same time to conduct a pool analysis, a meta-analysis, to be able to provide an evidence that could be stronger. And in this case, we weren't able to do it. Also, we haven't found in the literature any threshold that could tell us, okay, it is accepted from here to there, or this is a sensitivity that is accepted in the results of diagnostic utility for these tests. And at the same time, as has been said earlier, this is it's more complicated because these techniques start adding AI software, not only to sum up the power of the analysis, but also to show the components of the results. And as has been said earlier as well, the question more specifically was about the diagnostic utility as a starting point, and the idea to submit these metrics and the performance of these tests is just not sufficient. And this is something that we also wanted to convey. And this is the end of my presentation. You can find all the report in this QR code in our website. And I'll be standing by for your questions. Thank you so much, Rosa Maria. It's been really interesting. We'll certainly have lots of questions. So remember, you can write your questions down there in the Q&A section at the bottom of your Zoom menu. You can ask in English or Spanish. I can see that we have some comments. We'll certainly be receiving more questions. And I want to invite our next panelist, Dr. Virgilio Fayol Rojas. He is an evaluator for the functional unit of HDAs of the National Institute of Neoplastic Diseases in Peru, INEN. Virgilio, go ahead. Thank you so much. I'm going to share my slide. Give me 1 minute. Okay, let me know if you can see. Yes, it's perfect. We can see it. Okay, so good morning, everybody. I have a few minutes to be able to present. I want to take the freedom here to show you a study case, but also give you a few minutes to show you about our lessons learned and the evaluation of HTAs in the IVD and INEN. To give you some context, INEN, I-N-E-E-E-N, it's the National Institute of Neoplastic Disease in Peru, located in Lima. And 5 years ago, we created this health technologies unit. This unit of technology for these 5 years has been working in a dynamic changing of reports where we've been able to show variations, but this is the improvement for transparency and decision making. So putting you into context, this is what we're going to talk about. The use of the polymerase chain in real-time PCR for the for the HPV detection in cervix cancer, in cervical cancer. So I want to show you a little bit of the methodology and the processes that we follow, which is a presentation of the background and introduction, a context where we inform about the technology, the definition, the document where this reference comes from. And we use a methodology that, according to the approach, is that we're able to create a TICO question. We have to do a little bit more with health technologies based on that kind of performance, and a PICO question for a clinical approach. And so based on those questions, we establish a search in literature the widest possible, and according to that search, we make a selection of studies. And in the specific case of PCR, what we did was a search for papers where we ended up keeping 4. We had 4 selected studies. The first one was a systematic review from Lini in 2013, adding 3 clinical trials that were submitted after this review, where we're going to have the opportunity to show you further detail later on. So we showed our reports according to the evidence. We also ran a cost analysis. We have a cost presentation per se, but the office from the budget department helps us conduct the most effective analyses with a good budget impact analysis in it. Some administrative and organizational elements are also added there pertaining to that technology. So if the implementation of the technology to be used will demand some auxiliaries or accessories, then we have to measure that it's not causing an extra impact to our our budget, our estimated budget. We also attach annexes on the evidence assessment according to the criteria that was applied, either if it was Cochrane or clinical trials or AMSTER criteria, the Ottawa for the study of cohorts, and so many other things. And also, and this is to also put you into the context of what we've seen, we bring to consideration something to us that's very important, which is the efficacy. That means if the device is not only able to identify, but also to identify well medical conditions. So, in more specificity based on scientific evidence. So we know that medical devices, and most specifically, will have low evidence. And when we say low, we mean the number of studies that support it, and low also in quality. So it means that there isn't sufficient information to refer to from other experiences in other institutions. And so when that happens, what we do is we create a conservative scenario, and most particularly we prevent an indirect evidence, as we've seen with the in vitro devices. So we find about this direct evidence, quantity and quality, but in the case of the use of PCR for the diagnosis of HPV in women where we have a suspicion of a cervical cancer, and for the general screening as well, we're going to find the use of PCR that had the same sensitivity of the hybrid capture. But what happens here, we had a greater specificity, and most particularly, it wasn't the only point that we consider for that. There were diagnoses and experiences from other institutions that were saying that by using the PCR, they were obtaining what we want, the information of oncogenic serotypes, AC18 of HPV. So that was an interesting plus. It showed a larger efficacy in that new technology that we were going to incorporate. In terms of efficiency, we mean by that the device needs to benefit several healthcare programs. And what does that mean? Well, that technology is going to allow a faster clinical decisions that are also a lot more accurate. So with the resources that we have, with the incorporation of an IVD test, it's not only going to help us look into specific scenario or maybe for a specific service, but it's also going to help us use it for other programs. And it's served for like the Department of Cytopathology. It was useful for the Department of Gynecology, for preventive care, testing that are annually conducted for medical campaigns, for doctors in general, and other techniques as well that would allow a faster and more accurate diagnosis. In terms of equity, this device is providing benefit to different populations with high pathological burdens. We know that cervical cancer is very frequent in countries in development such as Peru, so it was meeting that requirement, and also it will benefit populations that had difficulties to access medical care. So there are several ways to capture info in what INN is looking toward right now is creating an interinstitutional agreement with other hospitals to be able to collect samples from even farther areas of coverage in the country in order to process these samples at the institute. So it allows a better expansion to those populations that have a difficult access to medical care. So this is a device that can be of benefit for everybody. Sustainability. It means that the incorporation of that technology is not going to compromise the healthcare system capability to finance other relevant interventions. So this technology wasn't even used in the 1% of the estimated budget cost. So the budget impact, as I mentioned, is an analysis conducted by a budget department that I told you earlier. Okay, now because of time, I'm going to go real quick. So during these years, we've issued 14 reports about 9 technologies, and you'll see that some have been implemented, some are pending for budget impact analyses, because without the impact analyses, we cannot conduct the given implementation, and some have been actually removed, like a serologic test for chemiluminescence for the screening of COVID-19. As we know, the beginning was really important, but this was now post-vaccination, and now there was incorporation of new technologies, and in time this was removed. Now there's a case here of the polymerase chain reaction for a case of HPV. We know HPV is paramount for the development of uterine neoplasias, and they seem to be high degree of malignancy as well. So the most important ones are PCR in real time and CH2. The diagnosis PCR has the advantage to make a difference of high degree oncogenes, particularly 16 and 18, because not all patients are going to end up in an epithelial injury. There has to be a follow-up. So literature, as we've said in our report, was based on a systematic review in 3 clinical tests. So, in summary, we found that PCR had a higher sensitivity and specificity in terms of hybrid capture. Both tests had a concordance, but it was about checking the oncogenic and non-oncogenic types, which is important for the follow-up of the patient. In spite of that, we applied the value testing because with PCR, we could find larger populations being benefited. It was faster, particularly for a population of 4,000 women to respond to that, considering it's not the only pathology found. So it was important, and the panel decided to approve of it. That's why that after the implementation, I want to be, and I want to emphasize this, the cytopathology department manage its resources properly. That's why right now we are caring for more than 4,000 women a year. And what did this department do? And I want to highlight here that it's not only about showing a report. We have to assess what does the department do. And as we see here, the space to be able to set the tests that are collected in different services. You go to different departments. You also have prevention campaigns being conducted and capture the samples and place them as you see here, having a space given for the amplifier equipment. They created a report template, which is what is issued at the moment of receiving a result, either negative, as we see here, positive. They conducted their legal documentation, but also they requested an external assessment. Hence, they have the approval from the College of American Pathologists for the department. So, in summary, technology has allowed to advance in these quick and testing, and the coverage of patients for these diagnoses and follow-up was also expanded. And not only that, but the department was able to integrate or to request an additional test, which was liquid-based cytology for such patients that were positive for HPV. So now they're conducted a liquid-based cytology to be able to detect what is the degree of injury, and it's a lot more recommended. And it's also indicated in some post-treatment guides. This, in the end, and as a summary, is that the challenges of implementation will also have logistic considerations and logistics that also involves human resources, space resources, and budget resources that need to be analyzed timely. And some final items here is, I need to mention that the lack of evidence in this quality studies is a limitation. Hence, we recommend to have a conservative scenario. And so when we have evidence, we need to measure if they have these value frameworks. And we also need to consider something, that technologies are constantly evolving. That is why that in the future, it is very possible that these technologies may incorporate artificial intelligence, most likely. And then once again, we have to keep an eye open for new information and making new decisions, always in pro of patients. That'll be all from me. Thank you so much for your invitation. Thank you, Virgilio, for the presentation and for sharing the experience of INEM. And we have a couple minutes more, and so we're going to go straight to the Q&A session. And I want to invite our panel members to open your cameras so we can have a conversation as a group. And I'm going to look at our chat. I see we have some comments about Virgilio, if you could put the full text of the paper, and Johanna asking for a reference of the paper as well. I'm not sure if she refers to specific speakers, so I want to invite all speakers to share your links to papers for studies that you refer to. Please put those links in the chat section so that all participants can have access to those documents and make their reading. I find that there is a comment in English from Gabriel Ogata.
Thank you, Stephanie. Yeah, there was a question about microcosting as kind of more appropriate approach to cost the whole patient journey, given the comments and what we've heard from the panelists about indirect and direct evidence, and obviously the impact of diagnostics. So I think, Chris, you've already added your reply there. Would you like to add anything else or summarize your view?
No, I think it's a very good point, that it's important to consider the whole clinical pathway. I think that's very important when we're looking at the claims of a test. I was just indicating that sometimes we have to be a bit practical about this. It can be very time consuming, particularly if you're using microcosting approaches to microcost the whole patient journey. So it's sensible to consider, when you're thinking about the claims of the tests and if there are aspects of safety, which aspects of cost might be most affected, and to focus on those.
Thank you.
Thank you very much, Christopher. I see, veo en la caja de preguntas y respuestas.
I see in the Q&A a question from Lida Osorio, and she says, in the diagnostic test evaluation terminology, there is a lot of variety. In these presentations, we find diagnostic utility, diagnostic efficacy. Are they the same? Do they refer to the measures of diagnostic performance, specificity, VPs, SNs, LRs, agreements? So I leave that question open. Okay, I can start. As you say, Lida, there's heterogeneity and the definitions. The efficacy is more a term that talks about therapeutical interventions. So the difference will be in clinical outcomes for the patient. And in general, refers to what you say, the operative characteristics, diagnostic precision, etc. But also, it sometimes incorporates safety or adverse effects of tests. So utility is more like an umbrella term. It's being used a little bit, I'd say a bit light, like in our documents in HTA that the agency uses. I think we use that term, diagnostic utility, trying to cover there both the diagnostic precision, sensitivity, specificity, predictive values, etc., also as with the safety there and other aspects. But these are a little bit of let's see, light terms sometimes. Sure, if I can add to what Federica said, that's why when I was saying diagnostic utility, I myself wanted to point it out what it was being requested, but also to be more pedagogical about it and teach what it means from the beginning. So the managers in that precision medicine unit were asking for something specifically. So they wanted the diagnostic utility in their case, like before being able to tell what's the impact, to be able to think about the clinical impact. And so for that, you previously have to know what's the performance. So we use the whole term of diagnostic utility, and we keep it for the performance of these tests. And then we say, okay, but this is the beginning. After that, you have to look at what's the impact this has, as it was being said, of the clinical pathway. So you can have a test that has a certain sensitivity, let's say 85%, but that's only the starting point. Now, what to do with that? And what is going to be the impact? So don't just stick with that. And so it's about doing that pedagogical approach with them as well. So when we teach them about what diagnostic utility means, we accompany that with a glossary also to be able to look into what are the expectations. You're looking for that. I'm going to tell you about the results. And I'm not going to say how many cancers are we going to prevent by a diagnosis of this, what's going to be the impact of mortality. These are other results. As they were saying, with the intervention and clinical impact. That is why it's important to be clear on that. And also the methodology for those systematic reviews, which is also different. Thank you, Federico and Rosa Maria. It's an important point there to make a little bit more precision about that particularity. Yes. I found another question there in our Q&A.
Yeah, we have another question about the key criteria when performing HTAs for in vitro diagnostics in your own context. And I would like to hear from all panelists if possible. I guess we only have a bit of time left, but if anyone wants to try and address the question from our panelists.
Well, if I jump in our case, we use a single value framework for rapid HTAs in all our documents. Now they are really very short-term documents, maybe similar to Virgilio's experience in NN. And our dimensions we assess three main dimensions. That is the quality of the scientific evidence, the net benefit, that incorporates harms and benefits, etc., and economic aspects as our main three high-value dimensions. It's hard to fit diagnostic tests sometimes in these dimensions, but that's our routine framework, if you want.
Yeah, and I guess that's probably a challenge for most value frameworks, because the reality is that you won't find evidence across all dimensions, or this is kind of like across the board an issue. We have another question, Stephanie. Should we take that? I'm mindful of time.
I think we can maybe one last question before we go to the closing remarks. So we have a question from Cristóbal Cuadrado, who's asking, la utilidad diagnóstica se relaciona con las
diagnostic utility is related to the properties, the performance, the diagnostic performance of the test, but also the way to apply that diagnostic strategy in the world of screening. This is very clear. Now, how do all the panel members link the evidence you show with the HTA strategies and experiences in screening strategies? And it was also an open question here for any of the panelists. The screening strategy is already an intervention. And so within it, we have an assessment framework, which is the framework document for population and screenings, where one of the questions talks about the diagnostic utility of the test you're going to use. That's one of the questions that you need to respond about this test, which is a selection test to be able to conduct the screening. But it's different and that part of the intervention to see what is the effect of that intervention. But at the same time, these parameters are going to tell you about the cost effectiveness model, the sensitivity analysis. So if we were to increase the sensitivity of these diagnostic tests, what would be its impact? How many people would you be able to serve to? And it's all linked. So you run the analysis of that test first, and then you measure what is the impact that it has in all that population or screening strategy. And it is a different question. And so it responds about this test performance to one of the questions of our framework document. But a screening is a whole lot more. It's a whole intervention that needs to be analyzed. If you allow me to just add something to that question, and it goes very much related to what I talked about, is that in terms of the screening, we also assess, and particularly the PCR diagnosis helps us a lot to implement not only for gynec oncology, but for preventive care that NN conducts. This was implemented in, and so this is part of the beginning of when it had to be budgeted. And as you said, when you have the diagnosis, then what's next, what you do. So it's not only about bringing up a diagnosis or go to the literature and they're going to tell us yes or no, you have a disease, but what you do next. Well, you have to have a strategy to give the proper care, and that's where the cytopathology department in INEN has analyzed in coordination with other departments to be able to have better coverage in terms of efficacy for the treatment and a good follow-up. Not all of them are going to end up in a diagnostic and then in treatment and then receiving therapies. But the idea then is to have a follow-up to be able to rule out whatever diseases we find. Thank you. Thank you, Virgilio and Rosa Maria. I found the last comment in our Q&A. Maybe I just hand it over to Anastasia for that last comment, and then we'll be able to close our session.
Yeah, thank you. It's a question from Henrique about the performance of diagnostics tests being impacted from obviously the composition of the eligible population. So I would like to know more about that. Is there a clinical and economic impact on different populations for the same diagnostic test? Don't know if any of the panelists would like to try and quickly answer that point. I think it's definitely applicable for screening populations. And
yes, well, yo creo que el sistema GRADE, por ejemplo, the GRADE evidence.
Yes, I find, for instance, that
tries to map this pathway, no, as the benefit, as for example, translated the clinical pathway to relevant outcomes, including mortality in cancer, etc., and adverse effects and false positives that in screening tests, for example, are really important and worrisome for a lot of patients. And this can be analyzed in different subgroups. No? So you have maybe average risk patients, and you can assess the outcomes in the average risk patients, or maybe in women with younger than 40 years old in mammography, and 40 to 50, that are different groups with different baseline risks. So you can assess these outcomes in different clinical pathways for different subgroups, no? To answer not only the main PICO questions.
Thank you, Federico. Chris, you want to add something?
Yeah, I'm a little bit worried about that as an answer or that as an approach. I think population screening is very different to tests used for diagnosis. And I would have absolutely no hesitation, having worked in both NICE and the National Screening Committee in the UK, to say if you're seriously contemplating a population screening process, you must have direct evidence. You must have randomized controlled trial evidence. It's very difficult to model it. I'm not even sure I really believe that modeling is really giving us that much better answers, even for diagnostic tests, but certainly for screening. The only place for modeling is really to look at the cost effectiveness, not to demonstrate the effectiveness.
Thank you, Chris.
Yeah, I agree. Mm-hmm.
Me too.
Thank you, Christopher. Muchas gracias todos.
Thank you, everybody. I see no more questions. And due to time, we're going to go to our closing remarks. I want to thank for the panel members for your time and contributions, and all participants in this conversation. I feel it is a very important topic. Questions will continue coming, and we'll continue working with these. I also want to thank Titai and the whole organizing team behind the screen. Thank you, everybody. We want to remind you that this webinar will also be available in the website of RedETSA and the YouTube channel of PAHO for the following days. And we also want to remind you that the annual meetings of the SHT will occur in a few weeks in June. So we want to invite you to participate. So thank you so much. And I now hand it to Anastasia for the closing remarks.
Thank you, Stephanie. Thank you to the panelists for their time and all the work they've done on the field, and all the attendees. Given the questions that we've got, I felt like, you know, we could go for another hour just kind of discussing the topic. So hopefully we're going to have a follow-up with some more of the specific questions in the next few months. So again, thank you, everyone, and enjoy your weekend when you get there.
Bye bye. Have a nice weekend. Buen fin de semana.
Que vaya bien, adiós.
Leo, muchas gracias.